Hello, and welcome to today's webinar, Quantitative and Systems Pharmacology Approaches for the Development of Oncology Drugs. My name is Suzanne Minton, and I'm a marketing specialist at Certara. If you experience technical difficulties joining the WebEx session, please dial 1-866-229-3239 if you're located in the United States or Canada. For all other countries, please dial 1-408-435-7088. To eliminate background noise, all phone lines will be muted for the duration of the event. Next slide, please. Please feel free to ask questions at any time during the event by clicking on the question mark icon on the floating toolbar located at the top of your screen. Please be sure and address your question to the default selection in the Q&A so that it goes to all panelists. We will address as many questions as possible during the Q&A session following today's presentation. This event will be recorded. And a link to a replay of today's webinar will be available on the Certara website within the next few days. Next slide, please. Before we begin today's presentation, I would like to introduce today's speaker, Dr. Karen Roland-Yeo. Dr. Roland-Yeo is currently a senior scientific advisor and principal consultant at Simcyp Limited, now part of Certara. Since joining Simcyp in 2002, she has supervised a team of senior scientists who have been involved in projects relating to the extrapolation of in vitro data to predict in vivo pharmacokinetics in humans. This has included development and implementation of the models into the Simcyp Simulator, which links the processes of drug discovery and development using simulations in virtual patient populations. During the past 6 years, she's been heavily involved in the consultancy projects relating to the application of the Simcyp Simulator within the pharmaceutical industry. Karen received her bachelor's degree in physics from the University of Natal. She completed a PhD in drug metabolism, a postdoctoral fellowship in the area of childhood leukemia, followed by a 5-year lectureship in clinical pharmacology and therapeutics at the University of Sheffield. She's had a very prolific career and has published more than 60 peer-reviewed journal articles. Karen, welcome to today's event. I'll now turn it over to her to begin today's presentation.
Thank you very much, Suzanne, for that introduction. So what I'm going to do now is just show go through the outline of the talk. And what I'm going to do is actually give you an introduction to PBPK modeling. I'm going to talk about system and drug-related parameters. I'm going to talk about how we integrate inter-individual variability. I'm going to then discuss the application of PBPK modeling in drug development, and then go on and describe a few case studies involving oncology drugs. But again, this is related to small molecule. So why are we interested in PBPK modeling? If you have a look at this diagram here, on the right-hand side, we've got the PBPK model components that consist of the system components. And then on the right-hand side, we have the drug-dependent components. And obviously what we're really interested in is actually looking at the intrinsic and extrinsic factors and how they can affect the system components, such as the blood flows and the different organ flows and enzymes within the individual organs. And by doing that, what we can do is have a look how these different intrinsic and extrinsic factors affect the system components, which actually then changes the PK or PD of the drug. And that is really why we're very interested in PBPK modeling at the moment. And also the other thing to say as well is that with respect to the PBPK models, they're moving models, and as we actually generate more data, we can feed that back into the model, and this leads us on to the whole predict, learn, and confirm paradigm. So before I actually go on and start talking about PBPK modeling, there have been a number of publications over the past year indicating the number of submissions that have been done to the regulatory bodies, and this was published by Xumei Huang in the Journal of JPharmSci last year. And on this particular diagram here, we've got the submissions according to the area of application. And you can see that the main area of application remains DDI, but importantly, pediatrics and also other special populations, including organ impairment, are also being represented in the submissions as well. Also, I think very important is that what we're showing on this particular slide here is if the sponsors included PBPK as part of their IND, that's shown in the red. In the green, it's if it was part of the NDA, but also, very important, is that even if the sponsor didn't submit PBPK as part of the submission, the reviewers actually developed models and then went back to the sponsors with additional questions. So the take-home message from this really is that if you're not considering PBPK, the regulators certainly are, and they'll come back to you with questions. I think, interestingly, some of you may have been at the FDA workshop about 2 weeks ago where they were discussing the submissions with PBPK, and even though on here it indicates that maybe about 30 submissions were done, I think it was anything between 50 and 80 that were done over the past 18 months. So there is a significant increase and therefore does need consideration. So going on to PBPK modeling, as I mentioned previously, we have the systems data where you have the different tissue components, blood flows, organ volumes themselves. This doesn't represent all of the systems data that we have in Simcyp, but certainly a lot of it. In the middle, we have the drug-dependent data, so we would like to have physicochemical properties, idea of the protein binding or the blood-to-plasma ratio, and importantly to have in vitro data determining the metabolism, permeability, transport if it's involved, and solubility if you're interested in the formulation side of things. And on the right-hand side, importantly, we would incorporate the trial design components as well. Now, all of this data gets put in together and scaled. We use in vitro and vivo extrapolation and generate the organ clearances and the organ-related data. And these get fed into whole-body PBPK models so that then what we can do is go on and predict the exposure PK in the population of interest. Now, I've got PD in brackets here. We do have PD modules, and therefore it is possible to relate the tissue concentrations directly to the response. But I'm not really going to talk about this in the webinar, but if anyone's interested, we can certainly follow up on that afterwards. So I mentioned that once we scale the in vitro data, we put it into a model, a PBPK model, so that we can predict the exposure. But I think importantly, and this received a lot of discussion at the FDA workshop recently, about how sophisticated the models need to be, or in fact operational models were actually mentioned. And what we mean by that is that you can see that on this particular side here, you've got the full PBPK model where you've got all of the different compartments. But on the left-hand side here, we have a lumped minimal model that can also describe the disposition of a drug. And it really depends on how much information you want, whether you're interested in the tissue exposure or whether you're just interested in the plasma exposure, as to whether you actually use a limp model, minimal model, or a full PBPK model. Similarly, we can describe the absorption by a first-order process, or if we're interested in looking at the regional distribution or the regional metabolism or PGP distribution, then we would need a sophisticated model like the ADME model that we currently have in Simcyp. And of course, if you're interested in looking at the formulation side of things, then you would definitely need to have this model. So in other words, what I'm trying to say here is that one of the key things about model selection is you have to, what is the question that you're trying to answer with the model? And then you make your selection based on that, or the level of data that you have available, or the intricacy of the model that you actually want to apply. So I've mentioned that we're very interested in incorporating population variability, and you can see on this slide here that if we want to consider the genetics, disease, environment, these are all very important issues that can certainly lead to differences in exposure in the population, and that is something that is the cornerstone of what we've been trying to achieve at Simcyp over the past 12 or 13 years. Now, how do we actually go on and do that? Obviously, it's important to incorporate the complexity of the covariates. So what we would do is select the population that we're interested in, and then this particular case, obviously, we're going to be talking about oncology. But the first thing that we need is the age-sex distribution in that population. And then what we would do is relate all of the other parameters to this. So say, for example, in a specific population, we would have a height-to-weight relationship, we would have a weight-height relationship, calculate the body surface area, and many of the parameters in the body relate back to these parameters. So we have these covariate algorithms within Simcyp, and would generate the appropriate parameters dependent on the actual population. Now, also, for a full PBPK model, we need to have the organ tissue distributions, and these have been collated from data from databases wherever available. And so what we would do within Simcyp is generate the individual based on the previous slide and then assign a size, so lung size to them, stomach size, liver size, heart size. But obviously, as I mentioned, it's important that we generate the appropriate size, because what we don't want to do is assign a large liver to a very small person, or in fact a pediatric subject. So this brings me on to Monte Carlo versus correlated Monte Carlo. If we have a look at this subject on the left-hand side, you can see that if you have, if we just randomly assign liver organ sizes to an individual, you could end up with an individual that has much larger organ volumes really than warrants the size. And that is why what we do is we use a Monte Carlo, a correlated Monte Carlo approach, to make sure that we assign the correct sizes to the individual, including adults and pediatric subjects. And this is what we mean by a correlated Monte Carlo approach, because obviously within Simcyp, what we're doing is we're inputting all of these distributions, and we use Monte Carlo simulation to assign all of the different physiological parameters to the individuals. And it's important that we put the covariates in, or the correlations in where possible, so that we end up with appropriately sized individuals. Now, what I'm going to do is, just after that brief introduction about how we, the input parameters that are required, the different models that we have, and also also how we incorporate variability, what I'm going to do now is just present two case studies. So this is drug Y. It's being developed for the treatment of various cancers. It's mainly metabolized by 3A4 to a major metabolite. During the in vitro data, it was noted that there was an IC50 shift, and therefore this was actually when the parent drug was actually included in experiments, and therefore it was important that the CYP3A4 inactivation parameters were determined, because obviously these are required to look at the time-dependent inhibition. So obviously, because of these, the implication of this, there's a potential risk of interaction with CYP3A4 substrates through mechanism-based inhibition of CYP3A4 by both drug Y and M1, because M1 was also a potent time-dependent inhibitor of CYP3A4. The other important thing is drug Y is likely to be co-prescribed with Everolimus, which is a CYP3A4 substrate, and therefore the sponsor wanted to know what is the predicted DDI between drug Y and Everolimus. Now, importantly, for PBPK modeling, generally when it's applied in the drug development process, PBPK models are developed at the discovery stage in preclinical species. These could then be refined and applied to healthy volunteers. And then importantly, the system parameters would be modified to accommodate the exposure seen in patients. And importantly, what I want to explain here is most of the two examples that I'm going to be presenting now, we're going to be starting with the PBPK model in healthy volunteers. And I think this is a very important point that was brought up at the FDA workshop meeting from a couple of weeks ago, but also very recently during PBPK presentations last week at the ASCPT meeting. It was made very clear by the regulatory groups that it is important to get the baseline model in healthy adults correct first before you go on and have a look at the exposures in special populations. And therefore, this is a starting point for us, developing the model in healthy volunteers and then seeing what we can do with the system parameters to look at the exposure in oncology populations. And obviously, we have to do this with Everolimus and also drug Y and M1. So the first thing to do was we had to develop a model for Everolimus, and these are the input data that were actually taken from the literature. I am aware that Everolimus is a PGP substrate, but in this particular case, the sponsor's drug was not an inhibitor of PGP, and also there were no data to incorporate into the model. So even though I am aware of that, I didn't build it into the model, but mainly focused on the CYP3A4 metabolism aspect. So those were the data inputs that were collated from the literature. And obviously the first step we want to see is how well we predict exposure in healthy volunteers. So I used a first-order absorption model and the and recreated the subjects that were actually used in the clinical study. And this is showing you what the concentration time profile looks like here. And on the right-hand side, there were two studies that I used where a 2 milligram single dose of Everolimus was used. And these are just the individual trials that I generated within Simcyp, and these are the population values here. And what I've done is actually included the observed data at the bottom, but you can see in terms of the Cmax and the AUC ratios, we're predicting reasonably well. So this is just looking at the everolimus exposure on its own in healthy volunteers. Now, we're wanting to use this as a victim drug to actually assess the TDI potential of drug Y. So what we have to do now is actually show how well the model works when we actually use it as a victim drug against time-dependent inhibition. And this is a model that we would have in Simcyp, where you've got the enzyme. If you have a competitive inhibitor or substrate binding, we always get the enzyme released. However, in the presence of time-dependent inhibitors, this would actually lead to accelerated deactivation of the enzyme, and therefore would obviously impact the exposure of the drugs. So this is what we had to build into the model. And this is actually just showing you the results of the prediction with erythromycin. So for this particular graph, what we've done is simulated multiple doses of erythromycin that is obviously a time-dependent inhibitor of CYP3A4, and then everolimus was co-prescribed, and we're looking at the change in exposure. And obviously on the y-axis, you can see that we've got the AUC ratio, and we've got the different simulated trials along the bottom here. So we've simulated 10 different trials, and what I'm showing you here is the median values and then the range. So this is the variability. This trial here is actually the observed data shown by the open circle, and what I'm showing you is that we're recovering the median value reasonably well, and certainly one or two of the trials are recovering the variability reasonably well as well for the predicted time-dependent inhibitor. So far what I've shown you is the exposure of everolimus on its own, and also a time-dependent inhibitor study with erythromycin. Again, this was taken from the literature. I have validated the model using rifampicin and other time-dependent inhibitors, but this is just to show you one example here. So now what we have to do is, this was actually done in a healthy volunteer population, so I've mentioned that we validated that. Now, in terms of oncology, I mentioned to you earlier on in the presentation that one of the things that we have to do when we're interested in a special population is get the age-sex distribution. So on the y-axis, that's the number of individuals. The blue is female, and the pink, the blue male and the pink are females, and we've got the different age groups here. So this was actually collated from study data, and we've derived this variable distribution and put those parameters into Simcyp. But what I want to show you here is, by having this age-sex distribution here, what this does by having those changes is this propagates through the algorithms within Simcyp. So in other words, we have a serum creatinine relationship with age, we have an MPPGL relationship with age. And MPPGL is mg of microsomal protein per gram of liver, and that scales to clearance. We would have a change in the cardiac index, cardiac output, height, weight. So essentially, the age would propagate through, and we would automatically get all of these differences in the oncology population. And one of the important differences that you would get by combining these parameters is actually a reduction in the CYP3A4 abundance of about 25%. On this next slide here, I also wanted to show you that obviously in patients, oncology patients versus healthy volunteers, we have a change in AAG levels, and of course the patient, the levels are actually much higher than healthy volunteer, and because the protein levels are higher, this can actually lead to a reduction in the fraction unbound of the drug of interest. On the other hand, if you've got, if your drug is mainly bound to protein, then obviously when we're looking at patient healthy volunteer values, the patient values are lower, and what this means is that lower plasma, you would actually get an increase in the fraction unbound. So it's very important. We've got these opposing effects going on, and it's not always easy to actually determine what is going to happen to the PK exposure unless you actually model it. So this is now showing you the prediction of Everolimus exposure in patients. So what I've done is taken the Everolimus model as it was. I've changed the age-sex distribution to reflect that of the oncology population, changed the protein binding, and now what I'm doing is showing that I'm able to recover the exposures seen in oncology patients at higher doses of 5 and 10 milligrams, as can be seen here. Now, so far, is what we've done is we've developed and validated the model for Everolimus in healthy volunteer and patients, and we've also validated the DDI prediction of Everolimus in healthy volunteer. And again, I'm reiterating this because, as I said, this has received a lot of discussion at the FDA workshop in terms of best practice about how to develop and validate a model. But obviously, everything that I've talked about so far has been with respect to Everolimus. But now what we need to do is actually develop and validate the model for drug Y itself and M1 in healthy volunteer and patients. And then what we did was applied the model prospectively to help with the design of the DDI study with Everolimus. I can't share you any of the details share with you any of the details with drug Y, but what I can do is show you that I used a lumped minimal model to actually reflect the disposition of the drug and the metabolite itself. And the metabolite was formed in both gut and liver, which means that as soon as it's formed, both of these moieties are actually able to inhibit the CYP3A4 enzyme. So what I've done is obviously develop the Everolimus and develop the drug Y and the model for the model for drug Y and M1. And also what I did was actually simulate these out at lots of different doses because I had the corresponding data, exposure data, over some single ascending dose and multiple ascending dose studies in oncology populations. And I've shown that we were able to recover the exposures over this dose range. So now what I had to do was obviously apply this model and use the model for drug Y and M1 with Everolimus, and this was actually used to optimize the study design for the clinical DDI. But what I wanted to show you is obviously what we have to consider is the auto-inhibition of drug Y and also the auto-inhibition of M1, the impact of M1 on drug Y on M1, but also importantly the impact of but also the impact of all of the moieties on Everolimus. And also what I haven't shown you here is that Everolimus itself is actually a CYP3A4 competitive inhibitor, and therefore we would have to consider the impact of that on drug Y and M1. So it is a complex interaction. But really, I don't have any results to show you, but what I wanted to show you was the process that we have to use to actually be able to go on and predict in an oncology population. So what I'm going to do now is show you the second example where I can actually show you more results and show you more of the data that we use to actually develop the model. So again, this is small molecule. It's a CYP3A4 substrate, a competitive inhibitor of both CYP3A4 and 2C8, and also both parent and metabolite, again, with time-dependent inhibitors of CYP3A4. Now, when I look at these, obviously what we have to consider is we have to develop a model to have a look at it as a victim CYP3A4 substrate, but also we have to look at it as a perpetrator as well, as because it's going to be a time-dependent inhibitor of CYP3A4, but also have a look at CYP2C8 substrates as well, and see what the impact is of inhibiting the CYP2C8. So this is the So what we've got here are the in vitro data for drug X, and we've had very good metabolism data for HLM hepatocytes and recombinant enzymes, and we were able to nail down the FM CYP3A4 for 70 to 80%. We had the fraction unbound for a range of concentrations, physicochemical parameters, CACO2 permeability data, and also inactivation data for CYP3A4 for both parent and metabolite. Now, one of the key things that I want to mention here is that the data were very, very robust for the in vitro side of things for drug X. Very good handle on the metabolism. The data correlated well from the recombinant to HLM and hepatocytes. And there I've mentioned that the FM was 70 to 80%, but actually you could bring it much closer to about 75% there. So very good data. These were actually the clinical study data that were available in healthy volunteers. We had single ascending dose, multiple ascending dose. There was also food effects, and also we had the all-important pivotal DDI study with Ketoconazole. Now, the reason I'm spending a lot of time on these two slides is, again, over the last couple of weeks and best practice, there's been a discussion about what in vitro data are required and what clinical data are required to actually develop and validate a PBPK model. And also, importantly, there was a big discussion about whether we need a DDI placeholder study. And certainly, in my view, we do. And this is why I'm spending a lot of time explaining the different data sets. So we had very good in vitro data, we had SAD data, MAD data, and we had a DDI study with Ketoconazole. So again, what I did was I had to consider the metabolite as well, because it was also a time-dependent inhibitor of CYP3A4. And so again, I used a lump minimal model for drug X and M1, and also have to form the metabolite in both gut and liver. But also importantly, and I have to make this very clear, is that when you're actually building in a metabolite, what we have to know is the formation of the metabolite from the parent drug itself, but also the disposition kinetics of the metabolite. So in other words, how is it cleared, and also the volume, and the same amount of information for the metabolite as parent, and that is very important. So again, we had to consider the time-dependent inhibition as well by drug X and M1, so we had to consider the enzyme kinetics. And this is actually now, what we're looking at here is the in vitro data for the drug, for the parent, and also the metabolite. This is actually looking at the exposure of drug X and M1 after a single dose. And this is quite a high dose, but again, this is actually in healthy volunteer individuals. But you can see certainly that we're recovering the exposure of drug X reasonably well, and also the metabolite, excuse me, the metabolite itself. And, but as I said, I mean, this was one particular case. Now, the other thing that I want to emphasize here is that this is actually a pure bottom-up approach. I mean, what we've been, there's also been a lot of talk about bottom-up, top-down. But the in vitro data in this case were particularly good, and when I put in the metabolism data, the fraction unbound, and the inactivation parameters, I was able to recover the exposure of drug X without actually refining using the PK data. So this is, and then of course what I had to do is obviously go on and actually validate further the model. So that was showing a single dose, but also we had multiple ascending dose data at 4 different doses. And what we've got here is drug X on its own and drug X during the last day of dosing. And obviously you can see here that this is a single dosing, but we're able to capture the accumulation reasonably well over the course of the study. But what is missing here are the exposure levels of the metabolite. And that is one of the issues that we had. We have metabolite data available for single dose at a high dose, but also we have metabolite data available at much higher doses in the oncology population. But in terms of validating the model in healthy volunteers, I only had the one set of data to actually work with. So this is showing you one dose. I've simulated out for all of the other doses and was able to capture the exposure over the dose range and also the accumulation ratio of the drug X itself. Now, I mentioned that, so what we've done is we've validated the model using single ascending dose and multiple ascending dose data. But then, of course, we've got that DDI study with Ketoconazole. So again, what I had to do was consider the auto-inhibition of drug X and M1, but obviously these are now in the victim drug positions, but also the impact of Ketoconazole on the metabolism of drug X and M1. So again, this is a complex drug-drug interaction where we're having to build all of these different components in there. And these are actually the results of the DDI study with Ketoconazole. So again, on the left-hand side, we've got the AUC ratio, and we've got the different trial groups going along here. And again, what we've done is I'm showing you the observed study data here, the closed circle. And again, you can see that in terms of the median value, we're capturing the predictions are capturing the observed study reasonably well. And also about 2 of the 3 studies are actually able to recover the variability that was seen in vivo. Now, again, I want to focus on this point because at the end of the day, this study is allowing us to confirm the FM value that we have for the actual CYP3A4 contribution for drug X. And that is why this study is very important in terms of the model validation. At the recent FDA PBPK workshop, it was mentioned, are we at the point now with CYP3A4 where we're comfortable with predictions, with ketoconazole for CYP3A4 substrates? And there was a lot of discussion on this. Certainly in my view, I still think it's a pivotal study to have, and because then what we've done is, in my view, we've cemented the FM value. We've validated the model with multiple ascending dose, but now this has allowed us to confirm the FM value. And now I'm actually happy with the model as it is to be able to go on and have a look in an oncology population, or in fact now have a look at the scenario where it's a perpetrator. Because what we've been talking about so far is actually we've been talking about using it as a victim drug. But the other thing to say as well is that, sorry, let me just go back to this. This DDI study with ketoconazole, what happened was this was actually multiple dosing of ketoconazole, but a single oral dose of drug X. Now, in my view, this was not the correct study design because obviously when you've got a time-dependent inhibitor, the normal thing to do would be to dose your drug to steady state and then actually give the CYP3A4 inhibitor ketoconazole. But what the modeling allowed us to do is to actually simulate this out. So if you have a look at this here, what we're showing is the active enzyme on this axis here, and we've got the time obviously going along here. Now, if you simulate just the drug on its own, this is what would happen during multiple dosing. So this would be what would happen to the enzyme level after a single dose. You would lose about 10% of the CYP3A4 active enzyme. But over multiple dosing, you would see that you would end up losing about 25%. Therefore, you're going to get a different result whether you dose whether you give a single dose of your drug versus multiple dosing. And you can see here that after a single dose at that particular single dose of the drug, the AUC ratio with ketoconazole is 4.2, whereas if you dose to steady state and then give ketoconazole, the value would be about 3.5. And of course, this is what happens when you actually give ketoconazole. You get a further reduction in the CYP3A4 level. Now, you may not say that this is a very big difference, going from single dose or multiple dosing, but obviously this is going to be dose dependent. And that's important because we've got quite a large increase in dose when we move from healthy volunteer to oncology populations, and therefore your susceptibility to the drug-drug interaction with ketoconazole is going to be very different. So just to focus on that a bit more, here we've got, so if we just look at the ketoconazole interactions here, so we've got a particular dose here. We're saying it's X of drug X, and then what we're saying is that if we increase that 5-fold and then by 25-fold, what actually happens to the drug-drug interaction with ketoconazole? And you can see here that at the lowest dose, it's 4.1. But obviously, when you give a 25-fold higher dose, because you've got much more of the enzyme being inhibited during auto-inhibition, the potential for DDI with ketoconazole is actually much lower. So in these types of doses, we're looking at healthy volunteer, but this is actually what's happening in an oncology patient, and therefore this is important. The other thing to say as well is that what I had to do was simulate out obviously strong versus, it's actually a moderate inhibitor here, and you can see that we definitely get the change, but obviously the dose dependence is less obvious because we're using a weaker inhibitor. Now, this is important, the strong versus the moderate, because obviously, I mean, there have been statements on PBPK labeling recently. And in this particular example here, there was sorry, let me just go back. There was this, what happened was that a sponsor developed a model, and then they had single ascending dose, multiple ascending dose data. They had the pivotal Ketoconazole study, and also a study with rifampicin as well. But what they were able to do is state that simulations using PBPK models suggested that moderate CYP3A4 inhibitors may increase the AUC by 6 to 9-fold. So in other words, because they had that Ketoconazole study observed and then were able to predict it, the moderate studies were actually waived, and a statement was able to go in the PBPK label. So this is very similar to the example I'm showing you now, where we had the SAD-MAD data, we've had the pivotal keto DDI study, and now, of course, what I've done is simulate out with moderate inhibitors and shown that you get a lower interaction. And this is a kind of scenario where we're certainly hoping that we can actually waive DDI studies and they can actually be included in the labeling. And so now what I'm going to show you is, so far what I've been talking about is actually using the model in the victim drug position, and what we were doing is looking at the impact of ketoconazole and fluconazole. Now what we're doing is, because obviously the drug X, it's actually a time-dependent inhibitor of CYP3A4, as is its primary metabolite, what we did was we used the model prospectively to actually help, first of all, design the DEI study with Midazolam, but then actually, and then of course the study's actually been, it's finished and we've got the results in, so it's great because it's able to confirm the model. But what I want to show you here is, again, 16 subjects were recruited into the study. The age, sex of the subjects were matched in the simulation, and here we've got the population values and the min-max. And on the right-hand side here, we've got the Cmax AUC ratios here. So this is really what I want to focus on here. So these were actually the observed data that came in, and you can see that here we've got very good overlap between the predicted Cmax values against the observed value, but also important with respect to the AUC ratio as well. We're predicting an AUC ratio of about 4.3, and in the population we were predicting 5.1. But what I want to show you, in actual all of the trial studies that were matched with respect to the number of subjects, you can see that we've got 7 studies that are within 25% of the actual observed value. So this really, really is, in my view, a very, very tight prediction. So as I've said, I mean, not only have we demonstrated that it's a good victim model, but now also in terms of the application of the model as a perpetrator, we've shown that it's also predicting very well. And even though I'm not allowed to indicate what the dose is, I can certainly say that this was at a much higher dose than has been used previously. So the fact that we were able to recover the prediction was certainly, in my view, a very good thing. So now, as we've said, all the discussion so far has been in healthy volunteer. And now, of course, what I'm going to talk about is application in oncology patients. Now, what I didn't mention with the previous example is that we know that, what the impact of cytokines is on drug metabolism enzymes. Cytokine levels are elevated in a number of inflammatory disease conditions, infections, and also in cancers. And decreased CYP activities, including CYP3A4, have been observed following exposure to cytokines in vitro. And if we have a look down here, this is a suppressed enzyme. These are the cytokines and cytokine modulators that actually have an impact on these particular enzymes here. We can see in vitro, this is a study that was done to actually have a look at the cytokine suppression of CYP enzymes. And on this axis here, we've got the 6-beta hydroxytestosterone formation, and here we've got increasing levels of IL-6. And you can see quite clearly that as you increase these levels, you do get a reduction in the CYP3A4 activity. And similarly, again, this is actually showing the IL-6 levels here in healthy volunteers, rheumatoid arthritis, and also post-surgery. And you can see that these values are very high. And if we look back to the previous slide where I showed you the dose-dependent relationship, with the IL-6 concentrations, you can certainly see that the values that we're actually attaining in the clinic are much, much higher than these, and therefore we certainly do expect a reduction in the CYP3A4 activity. Again, this is just showing the percentage of the population with this range of values and also post-surgery here. So the clinical consequences of increased IL-6. Well, really, I mean, there was a poster that was presented last year by Lilly at the ASCPT meeting, and there was a very nice study there where they looked at healthy volunteers and oncology population. And what was nice about this particular study was that they looked at the probe substrate Midazolam. And what they showed was that there was a 28% reduction in Midazolam clearance, and that was in cancer patients versus healthy volunteers. So we definitely know that it's important. There have been a number of other drugs as well, including simvastatin, where there's been a clear change in exposure. But what I wanted to mention here is that when you have patients with inflammation, infection, or cancer, they've already got elevated cytokines and IL-6. And so in these patients, there will already be a reduction in the P450 activity. However, when they're given a drug that actually affects or reduces these cytokines, then what happens is that stops the suppression of the enzyme. And then, of course, what happens is over time you start getting an increase in the enzyme levels. And what this means is if we have a look at this particular concentration on here, we've got plasma concentration. So this would be the cancer population here, where what's happened is they've got elevated cytokines, reduced CYP3A4 activity, and therefore they have increased exposure. However, when they're treated with a drug that impacts on the cytokine levels and stops the suppression of the enzyme, suddenly we start getting an increase in the enzyme, and therefore we get a decreased exposure. So this is quite complicated. So at the time of doing this modeling, I was aware of this and debated whether to try and put some function in here where almost like an induction function where I start increasing the CYP enzyme over time. But already, because the model was quite complicated with respect to the time-dependent inhibition of the drug X and also the metabolite, and also incorporating the other changes in the oncology population, I started off just going with a simpler model, but I just wanted to make you aware of these changes in there. So again, what I meant by that was, again, we would incorporate the age-related changes for the oncology population. And as I mentioned previously, this would automatically lead to a 25% or 27% reduction in CYP3A4 anyway, apart from actually the suppression. So that is why I didn't build a suppression model in there. And then, of course, we have to consider the binding differences. For this particular drug, it was actually bound to AAG levels, and therefore there was actually a reduction seen in the fraction unbound. And these values were actually measured anyway. So I was able to incorporate measured values rather than predicted values in the model. Now, this is actually showing you the model validation step. So what I did was incorporate, obviously, the age-related changes, the change in FU as a result of the change in the AAG levels, and simply used the model for drug X and M1 that I developed in the healthy volunteer population. So nothing has changed for those parameters there. But you can see that we are able to recover the exposure of drug X and M1, and this is, I have to say, is a very, very high dose, and therefore I was very pleased when we were able to recover this. If I hadn't have been able to recover it, then I probably would have gone back and tried to build in the suppression and to see how that changed. Because obviously what we've got changing over time here is a time-dependent effect, which is obviously decreasing the amount of enzyme. And then you've got the impact of the drug on the IL-6, and then obviously what's happening is the enzyme is increasing over time. But just to make it clear, I didn't incorporate those changes here. But certainly this is just showing you that one of the dosage levels in the oncology population over multiple dosing, and there were other doses available that I could actually use to develop, to actually validate the model. And again, I had very good predictions. So, of course, now what we have to do is have a look at the actual DDI study with ketoconazole. So this hasn't been conducted. But what we wanted to do is obviously apply the model in this particular situation. So we've got and what you can see is that you've got a single dose of the drug X. So this was actually the healthy volunteer study that was performed. where we're predicting a value of 4.1. However, if we increase now the drug and by 2.5-fold or 7.5-fold BID and give this in the oncology population, what we're actually predicting are values of 1.5 and 1.3, and therefore significantly lower than the values that were actually observed in healthy volunteers. But obviously, this is mainly related to this dose dependency here. And if we have a look at the bottom, what you can see is because, so this is actually the dosing at X. This is what I showed you before. But obviously at the much higher dose of 7.5X, you can see that you wiped most of the CYP3A4 activity out, and that is why we have this much lower potential for drug-drug interaction with ketoconazole. Okay, so I think that's all I've got to say on this. Except for I'd like to acknowledge the Simcyp team. I did most of the work for the actual consultancy examples, but certainly without all of the models and the other data, I wouldn't have been able to do it without them. So just to acknowledge the Simcyp team. And now I'm going to hand over to Suzanne, who's going to moderate the question-answer session.
Thanks, Karen. The first question is, you've shown two examples where clinical data has been used to help validate the PBPK models that have been developed. What is the point of developing the PBPK model in the first place if you still have to perform the DDI study?
Well, that question, that's actually a very good question. It came, we had a discussion on that actually during one of the PBPK sessions at the ASCPT meeting. And that point was made. And I think what was quite interesting then was actually Peng Zhao from the FDA got up and said that in his view, it's still important to have that pivotal DDI study to actually validate the model. So I think even though, and the other thing to say as well is that in one of the examples that I showed you for the labeling, one of the studies, they had to do the keto study, but then they were able to waive the other studies with the moderate inhibitor. So I still think, certainly in my view, I think it's a good thing to have, because then also, once you've got the robust model, you can apply it to lots of different scenarios for moderate inhibitors, but also special populations as well.
Your presentation was on small molecules. Do you also have PBPK models for large molecules?
Yes, we do, and we can certainly, and for those who are interested, we'll certainly be holding a webinar at a future date to explain the different models that we have for large molecule.
In your view, would the regulatory agencies accept simulations in oncology patients for the DDI study with ketoconazole at the much higher dose of drug X?
I don't know. I don't work for the regulatory agency, but certainly I think one of the problems that we have is that we're not able to conduct all of these DDI studies in these patients. It's just not going to be possible to do that. And I think as the DDI study for ketoconazole has already been done in the worst case scenario, well, single dose in healthy volunteers, certainly in my view, I think that that they may consider it.
And this will be the final question. This is a two-part question. Is the oncology population already in Simcyp version 13? And what are the changes compared to Northern European Caucasian healthy volunteer populations besides age, sex distribution, and plasma protein concentrations?
So, the main differences are, in fact, it's just the age-sex distribution. That's the main difference between the, sorry, was that the North European Caucasian and healthy volunteer?
Yes, it was.
Yes, okay, so that's the only difference there. And with respect to the first question, it is not available at the moment, but there was but I gave the reference Chiti et al. It was developed by Genentech, and they derived the variable parameters for the age-sex distribution and also the different AG levels. So no, it's not available yet, but it will be available, because what we're trying to do at the moment is have a look at the suppression side of things.
Okay, thanks, Karen. Next slide, please. A reminder that a link to a replay of this webinar will be available on the Certara website within the next few days. The current slide provides information on whom you may contact with questions you may have regarding Certara's products and services in the Americas, Europe, India, and Japan. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes today's webinar, and you may now disconnect.